Hello, this is Mr. London, and this unit we're going to be talking about heredity. That's the passing of traits from parents to offspring. So this is some of the vocabulary we're going to be using in this unit. One of the words we're going to be using a lot is this little guy right here, allele. That means basically that's a version of a gene or a trait. So different genes have come in different varieties, and those are called alleles. For example, earlobes come in two different shapes. There's the attached earlobe allele, and then there's the free lobe allele, okay, which I have. And that's actually the more common allele because that's the dominant allele. We're going to talk about why that is, what that means. So who was the first guy to kind of understand how traits are passed down and inherited from parents to offspring? This monk called Gregor Mendel. And he had a lot of time on his hands, and he did a lot of experiments with plants, and he was able to figure out a lot that kind of laid the groundwork for what we know today about how traits are inherited. And this was before we really knew about chromosomes or DNA. So it's pretty remarkable. But he was like the first one to use probability to predict how traits are passed down. Okay. And I'm gonna explain what that exactly means in a bit. But probability again is has to do with chance. So for example, when you flip a coin, there's a 50% probability you're gonna get heads, and a 50% probability you're gonna get tails. Okay, so that's what probability means. One key point I really want everyone to know before we go on is that organisms that reproduce sexually, like animals and plants, have two copies of every gene. One copy from each parent. Why is that? Because we have two copies of every chromosome. We get half a chromosome from one parent, and we get the other 23 chromosomes from our other parent. Remember, humans have 23 pairs of chromosomes, and for every gene we have at least two copies. Well, we do have two copies. Let's talk about Mendel's experiment. So he experimented with plants, and one experiment he did was he cross-pollinated purebred or pure white flowers with purple flowers. So when you cross-pollinate flowers, basically the flower is the reproductive part of the plant. It has pollen, which is the male gamete, and it also has the female part. So it has the female gamete. And when the pollen goes from one flower to another flower, it fertilizes the female part of that flower, and that it produces a seed. So anyway, he cross-pollinated white flowers with purple flowers, and something interesting happened. That second generation were all purple. He called that second generation the F1 generation. And he called the parental generation, that first generation, the P generation. Okay, so what happened to that white allele? It was gone. So again, what was it gone or what happened? So in order to figure out to continue with this experiment, he cross-pollinated the two purple flowers from the F1 generation. And then he got something even more interesting. The white allele reappeared. So in the third generation, known as the F2 generation, 75% of the flowers were purple, but 25% were white. So that white allele reappeared in that third generation. So what did he deduce from that result? He determined that white allele wasn't gone, but it was hidden or masked by that purple allele. So he called that purple allele the dominant allele, and he determined that the white allele was a recessive allele. What does that mean? Like I mentioned, for every trait, traits come in different varieties called alleles, and some alleles are dominant because they overshadow that recessive allele. And he kept getting the same result over and over again. every time in that F2, that third generation, he kept getting a 3-to-1 ratio where that recessive allele would reappear in that third generation in a 3-to-1 ratio. So let's go over some more vocabularies. These terms, homozygous and heterozygous. So remember when I mentioned that we have 2 copies of every gene or trait? If you have 2 dominant traits, so 2 identical dominant traits, you are homozygous for that trait. So you could be homozygous dominant, where you have 2 identical dominant alleles, or you could be homozygous recessive, where you have 2 identical recessive alleles. And before I explain what these letters mean, I want to explain that in genetics, instead of writing, for example, with flower color alleles, instead of writing purple, purple, we usually use the first letter of that dominant allele. So in this case, with flower colors, we're abbreviating purple by writing a capital P. The dominant trait is always written as a capital letter. The recessive allele, in this case, it was the white allele, is written as a lowercase letter. So little p represents white color, and big P represents purple color. So if someone has 2 big P's, that means they have 2 identical copies of that purple allele, and they're homozygous dominant. If they have 2 little p's, so they have 2 white alleles, they are homozygous recessive. Now, what if they have one? Remember, again, we have 2 copies of every trait, 1 from each parent. And if you carry 2 different alleles, so 1 of each, 1 dominant and 1 recessive, you're known as heterozygous. So if there's a big letter and a little letter, you're heterozygous. If there's 2 big letters or 2 little letters, you're homozygous. Okay? One easy way to remember that is that homo means same and hetero means different. So in this case, there's 2 different size letters, and homozygous, they're the same size letters. Okay? So again, just to review, guys, we've got for every trait, everyone has 2 copies of every gene, because we have 2 copies of every chromosome. So this represents one example. So this is one. These are plant chromosomes, and in this example, we have 1 homologous pair, and you have 2 copies of every gene or trait. So this flower has a purple allele on one of the homologous chromosome, and on its partner chromosome, the other homolog, we've got a white allele. Okay. So again, just to review, guys, some alleles are dominant and some are recessive. Dominant traits are more common because they overshadow that recessive allele. So remember, let's say you're heterozygous, right? You've got you carry that recessive allele, but you also carry the dominant allele. You're still this plant is still going to be purple because that purple allele is dominant, and that will overshadow that recessive trait. Even though this flower carries that recessive trait, the result is the same. It's still a purple flower. Okay, it's the same result as if it was homozygous dominant and carried 2 purple alleles, because again, the big letter dominates that little letter. Okay, so again, individuals with dominant alleles have the trait or have that dominant trait, whether you have 1 copy, like with this heterozygous case, or you have 2 copies, like the big PP. All right, guys, let's review. Plants that are blank produce offspring that look we're going to skip that one. No one cares. Different forms of genes are called alleles. Blank alleles overshadow recessive alleles. Dominant organism is blank when a gene, for a gene, when its cells contain 2 different alleles. Heterozygous. All right, next. The parental generation is abbreviated as the P generation. The offspring generation is called the F1 generation. Capital letters represent dominant alleles and lowercase are recessive. All right, great. Phenotypes and genotypes. Yay, more vocabulary. Genotype is the combination of letters. So it's the combination of alleles. All right. So, for example, eye color genotype of a heterozygous person. One big letter, one little letter. That is heterozygous. So if I asked you what is this person's genotype, it would be big A. You would say big A, little a. So the genotype is the combination of alleles. It's the letters. So this person's genotype is big A, little a. The phenotype is the physical characteristics for this trait. It's what you see. So this person's phenotype would be brown eyes, because big A represents brown, and that's dominant. So this phenotype would be brown. The genotype would be big A, little a. Okay? So a person that's little a has the phenotype green eyes. They have the genotype little a, little a. Okay? All right, great. So let's do some Punnett squares. Now this is going to be hard to teach in a PowerPoint because I don't have a whiteboard. So I'm going to try to use Jamboard, and we're going to see how that works. So Punnett squares are used to predict the possible genotypes and phenotypes of the offspring, which is what Mendel did. So how do you set up a Punnett square? So basically you put one parent's genotype up here, so one letter or allele here for that one parent, and it's the parent's other allele here. And then for parent number 2, you put that parent's genotype here on the left. Okay? Just like that. So these both parents are heterozygous. So again, this is the parent's genotype, and each box represents. So these are the boxes represent the possible genotypes of the children based on the parent's genotype. So if we wanted to know, okay, we've got 2 parents that are heterozygous. What are their parents? What are the children going to be like? We do a Punnett square, and each box represents 25%. So, and let me just show you what I mean. So how do you fill a Punnett square up? Basically, it's this A and this A, where they meet in the middle, fills up this box, okay? So you're going to drag this A down, drag this A over, and both those A's together make big A, big A. For this box, you're going to drag this A over and this little a, lowercase a, down here. And where they meet, you have a heterozygous combination. They combine to make a heterozygous genotype. And then you do the same for each box. So again, drag this big A down here. It's almost like tic-tac-toe. This little a goes over in this box, and when they combine, they form a heterozygous offspring. Okay, and the same with this. So this little a and this little a together combine to make a homozygous recessive offspring. So these represent the 4 possible genotype of the offspring, and each one represents 25% chance. So we've got a 25% chance of being homozygous recessive, 25% being homozygous dominant, and a 50% chance, because 25 plus 25 equals 50, 50% chance of being heterozygous. All right, now I'm just like an idiot. Okay, guys. So let's do some Punnett squares. All right, so tongue rolling, yes, it is apparently a trait, I guess. And it is being able to do it, well, that's a dominant allele. So being able to roll your tongue is a dominant allele. So if you're heterozygous, yes, you can roll your tongue. So let's do a Punnett square, okay? And little t means you can't roll your tongue, in case you like this Emma Watson person. I can't remember what his name is, but I know he's that witch. He's that witch from Harry Potter. Yeah, he's a witch because you ride the broom. All right, so if 2 parents are heterozygous for tongue rolling, what percent of their kids will be able to roll their tongue? All right, so let's do this Punnett square. So you can pause it and try to do yourself, and then replay when you want to find out the answer. Okay. So Those T's go across. You know what? Let's do this on a Jamboard. Okay, so they're both heterozygous. All right, so they're both heterozygous. So I'm going to put one parent up here and the other parent's genotype up here. Okay. So again, this big T is going to go down here, and where these two meet is in this box. They combine to form a homozygous dominant. Again, this big T is going to slide along to this box as well and combine with this little t to make a heterozygous. Big T down, little t over here. And while we're at it, let's bring this little t over here too. And then finally, this little t comes down here. And what we have is we have 25% homozygous dominant, 50% heterozygous, and 25% homozygous recessive. Okay. Now here's something interesting we haven't talked about yet. This is asking what the genotypic ratio. So in a ratio, you're comparing quantities. Okay. So so we have 4 possible outcomes, and we're going to count. There's 3 different genotypes. We've got, again, homozygous dominant, heterozygous, and homozygous recessive. So we have to show the ratio of each of the 3 possible genotypes. Remember, genotypes are the letters. So I like to put the homozygous dominant first. So we have 1 big T, and we have 2 heterozygous, 1, 2, to 1 homozygous recessive. So that is a ratio. You write it like that. So you put that's how you write it. Okay. I'm not going to over explain it. So phenotype. Okay, so this is a little bit trickier. So again, phenotype is what you see. So remember, big T represents being able to roll your tongue. So even though these two are heterozygous, because a tongue rolling is dominant, these 2 individuals are going to be tongue rollers just like this individual right here. So we've got 3 tongue rollers and only 1 non-tongue roller. So with the phenotypic ratio, with this simple type of inheritance, with dominant recessive inheritance, there's always only 2 possible phenotypes. Typically, you could okay. So you could have the amount of genotypes varies. In this case, we have all 3 possibilities. All right, great. So that is how you do that Punnett square. All right, let's move on. So okay, here we've got another Punnett square. Let me just go into present mode. We've got homozygous dominant and a homozygous recessive. Let's do this Punnett square. So again, little t again, not being able to roll your tongue. So what is the outcome? This is going to be the outcome. In this case, all 4 are heterozygous, and all 4 can roll their tongue. So what would the phenotypic ratio be here? It would be 4 to 0. 4 tongue rollers to 0 non-tongue rollers. And the genotypic ratio would also be 4 to 0. You could write 0 to 4 to 0 because there's no homozygous dominant and no homozygous recessive. But on a test or quiz, I would accept 4 to 0 or 0, 4 to 0 for the genotypic ratio.